Hello everyone, welcome back to my channel, the Biolab Scholar. So today I will be discussing what kind of content you can expect on the channel. So basically I have divided the content on the channel into three categories and I'll be elaborating on each of the categories. So the first category is about the lecture series. So in this lecture series, I will be covering all the basic introductory biology lectures from cell biology, molecular biology, genetics, and if I go specific into the topics, you can expect topics like cytoplasmic meal sterility, DNA replication, and enzyme kinetics, and etc. And I would like really appreciate whatever suggestions you would give, and just comment down below if you have like any requirements for any of the specific topics. And then the second topic is like really close to my heart. It's about advanced research lab protocols. This topic specifically is very close to my heart because when I started my PhD in Canada in molecular biology, I had a very hard time, and I think we should accept that whenever we have a protocol from any of the publications, it just doesn't work. Like it really gave me a very hard time. So I have to come up with some troubleshooting and tips and tricks to do a protocol. So I will be sharing all those tips and tricks with you in a series, and I will be covering different kind of techniques like molecular cloning, all the protein work, including Western blots, CoIP, and generating transgenics, how we design the um, uh, CRISPR-Cas9 construct. So basically, mostly I think it will be covering the molecular cloning because in molecular biology, the basis is molecular cloning. Uh, where like if you need any of the transgenic plants, like if you have to study a gene, we need like knockout lines, we need overexpression lines, we need transcriptional reporter lines. So all of these comes from the clones that you make in the in your lab, and then you use like different techniques to transform your plants. Like we have dicot plants, we have monocot plants, and then there are differences in the techniques how both the type of plants get transformed. So I will also be covering that kind of topic, like which technique is better for which kind of system. And then to make all those different clones, what kind of vector you should pick up, and how to make a certain clone. If you need to make a transcription reporter line, what to how to choose a vector, because there are n number of vectors, but what you should choose. Like you can get like lots of publications and mentioning lots of uh, constructs, but what is best for your study, it also depends on your experimental design. So I would be happy to help you in uh, that kind of planning. So I would like really appreciate if you can comment down below and ask if you have like any specific questions related to your study, your experimental design, and I will try my best to solve them. And the third category is about all the prospective students who want to come to Canada on a funded master's or PhD basis. So I will be helping you to find how you can apply uh, for a funded position in Canada, how to approach the professors, how to search for a professor, because it's it's a long-term commitment. Like really, like a research is a long-term commitment, and you should be like 100% sure before you get into a professor, and like uh, where you don't find that kind of comfort later. So it is very important to know uh, your research area, your supervisor's research area, then how to email them, how to get into touch, like what you can expect in the interviews, how to write emails, how to apply for visa, and then if you have like internal funding, that's great, but how to apply for external funding. So all these factors like really matters. So I will be helping you in all these matters. So these are the broad three categories. Like so just for a quick brush up. Like first will be the lecture series that will be covering all the basics of biology, and then second will be mostly research driven molecular biology techniques, and then third is to help all the prospective students to come to Canada on a funded basis, like a funded PhD basis. So yeah, so stay tuned. Do subscribe and share and like and comment as much as you can. Please help the channel to grow, and as the family grows, like we will all come to know each other better, and we can all expect better from each other, and I would be able to help you even better. So yes, stay tuned, stay healthy, stay happy, and see you in the next video. Bye bye.